Okay, it looks like the number of participants joining this webinar is starting to level off. So I think we will go ahead and get started. So hello, everyone. Thank you for joining us today for this second webinar in our EPIC series on the use of NAMs in risk assessment. This series is being co-organized by the Environmental Protection Agency, PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. I'm Rachel Bruner, a biologist with the US EPA, and I'll be co-moderating today's webinar along with several others, including Gino Scarano, a senior advisor from the New Chemicals Division of the EPA Office of Pollution Prevention and Toxics, Niladri Baumek, a toxicologist in the Human Health Assessment Branch of the California DPR, Amy Clippinger, president of the PETA Science Consortium International, and Christy Sullivan, the vice president of education and outreach at IIVS. Can advance to slide 2. So we have 2 speakers today who will be presenting on the development and application of NAMS workflows and tools. First, we'll hear from Dr. Alistair Middleton from Unilever, and then we'll hear from Dr. Tara Barton-Maclaren from Health Canada. Before I introduce our first speaker, I just would like to note that the slides and the recording from today's webinar will be posted online at the link on this. Should we go to the second slide? There we go. Okay, so the slides and the recording, they'll be posted online at the link shown here on the screen shortly after the webinar. You'll also find all future EPIC webinar materials will also be available at this link as the series continues. We will do a Q&A session at the end of the second presentation. The webinar is scheduled for 1.5 hours, but we may wrap up earlier, kind of depending on how long the questions go. Everyone is currently muted except for me, and you can type any questions in the questions module within your Zoom toolbar, and you'll see that at the bottom of your screen. And when you do so, please indicate if your question is directed to a specific speaker. And we just ask that you don't use the chat function just to make things easier and more straightforward. We have also enabled closed captions, and you can turn this feature on or off according to your preferences, also using that toolbar at the bottom of your screen. At this time, I would like to invite Gino Scarano to share some introductory comments from EPA.
Thank you, Rachel. Hello, everyone, and thanks you for joining us for this second EPIC webinar. I would once again like to thank the 2 of our co-organizers, PETA, the PETA International Science Foundation, and IIVS, for handling all the logistics, the website, and the handling of all the materials. And also including Dr. Baumek. I want to thank everyone on the planning group for helping to plan these series and invite our wonderful speakers. And I know the introduction will be coming shortly, but I'd like to welcome Drs. Middleton and Barton-Maclaren for what I know are going to be great presentations on how we can all think about using NAMs and incorporating them into our risk assessment. So, and it's good to see you, Tara. I haven't seen you in a while. So let's get started. Thanks. Back to you, Rachel.
Thank you, Gino. Our colleague from the California DPR, Niladri Baumek, would also like to share some comments now.
Thank you, Rachel. Hello, everyone. Once again, welcome to the second session of the EPIC webinar series. First, my sincere thanks to all panelists for including California Department of Pesticide Regulation to this EPIC webinar series on a very important topic on new approach methodologies. We are excited to be involved along with US EPA, IIVS, and PETA. And I look forward to great presentations from wonderful speakers. Best wishes to them. And I now return it back to Rachel.
Thank you, Niladri. So speaking first today, we'll go ahead and get started, is Dr. Alistair Middleton. Dr. Middleton joined the Unilever Safety and Environmental Assurance Center, or SEAC, in 2014. After working in academia as a research associate in Germany and in the UK, where he used mathematical models to understand the dynamics of complex biological systems, including hormone transport and gene networks. He is now a science leader in computational toxicology, where he leads a multidisciplinary team looking at how we can use computational models and in vitro data to make robust safety decisions without the use of animal testing. He has a Bachelor of Science in Mathematics from the University of Edinburgh and a PhD in Applied Mathematics from the University of Nottingham. Welcome, Alistair, and please go ahead.
Okay, thank you very much, Rachel. So I'd just like to begin by thanking the organisers for giving us this opportunity to present some of the work we've been doing on developing and evaluating a systemic safety toolbox for the use in next generation risk assessment. Hopefully the slides are moving forward. So for those who don't know, in NGRA is essentially an exposure-led and hypothesis-driven approach where you're looking to understand how you can integrate new approach methodologies within a tiered decision framework so you can conduct your safety assessments without the use of animal data. And a key hypothesis underpinning this sort of work is that if there's no bioactivity at the consumer relevant concentrations, then there can be no adverse health effects. There's been a number of different decision frameworks that have been published in this space, notably the CURAT-1 framework and also the EPA toxicology blueprint published by Rusty Thomas in this group. But these different frameworks form a similar structure in that they have a tiered format. So beginning at tier 0, you're doing problem formulation, so collecting the available evidence you have on your chemical and your exposure scenario, taking into consideration, for example, information you might get from various hazards and alerts. And then in many cases, you can already make a decision at this point. But then in some cases, you may want to go to a further level of refinement. So in this case, we have tier 1, which is where you would try and estimate a bioactivity exposure ratio. So that is essentially where you're taking the information about the use case and exposure scenarios that I just spoke about and running, for example, PBK models to estimate internal exposure levels, and then coupling that with data from in vitro cell assays that are intended to cover off a broad range of biological effects that the chemical might be causing that may be of toxicological concern. So the idea there is that you're then modeling that data to estimate points of departure. And so a key metric from the decision point of view is this, as I say, bioactivity exposure ratio, which essentially gives you a measure of how far the exposure is from points of departure. So if your exposure levels are very far below the PODs that you get, then in many cases you can already conclude that that is low risk based on that tier 1 assessment. But of course, taking into account whatever information you got from your problem formulation as well. However, in other cases, you might find that the exposure levels are close to the points of departure. So you've got a small bioactivity exposure ratio, in which case we would consider this to be uncertain risk. So you may be triggering some bioactivity, and then you can go to a further tier of refinement where you would maybe look to refine either your internal exposure estimates by, for example, generating maybe clinical data, or refining them further in terms of getting a better understanding of the biological effects that are being triggered by the cell in the different assays. By the chemical in different assays. So we published a sort of case study looking at this for a particular example where we considered coumarin used as an ingredient of various consumer products. This was published in 2020 in Barton-Maclaren Toxicological Sciences. So I don't have time to go into that, but essentially it was gave us confidence that we could indeed use these NAMs within that sort of tiered risk assessment framework to make a protective safety decision and understand how we can bring those different elements together. However, what we wanted to really do is then move beyond that and try and think about what would be a core suite of tools for that tier one component of the overall risk assessment framework. So for estimating that initial BER that you get that I was speaking about. So that would be essentially understanding what are the range of different in vitro cell assays and exposure models you should be using to estimate that BER, but also trying to understand, okay, once you get a number out from those data, at what point can you say confidently that something is low risk, or maybe whether a given exposure level is actually uncertain risk and you need to do further refinement. So essentially, what is that decision point? The third point here, I'd just like to emphasize, of course, that although we're focusing on that specific core set of tools, in practice, we would always be doing this within an overall tiered assessment framework. So we would always have information, for example, that we would gather during the problem formulation and take into consideration outputs from in silico tools and other pieces of information that would be used within the weight of evidence. So the core NAMs under consideration are, of course, the PBK models, in vitro pharmacological profiling, high throughput transcriptomics, and a cellular stress panel. And the idea here is that we're trying to select assays that can cover both non-specific and non-specific mechanisms of toxicity. And in particular, what we're trying to do is have assays that can detect the very early perturbations that may occur that may be triggered by a chemical that could potentially lead to toxicity, but before the onset of any adversity. So by doing this, the idea is that you're being potentially protective of toxicological effects that may occur over, for example, chronic exposures, because you're just trying to capture those very early stages of that. So once we determined what were the suite of tools that we wanted for the toolbox, we also had to think about what was the approach we wanted to do for really assessing it in terms of whether or not it's fit for purpose. So in the context of what we're talking about here, it's really, can you actually use that toolbox confidently to make safety decisions that are protective of human health? So as opposed to, for example, using it to, for example, predict some adverse toxicity. So it's really about making protective safety decisions. And related to that, of course, is that question of, do actually that set of assays that I just described, is that actually sufficient for providing that biological coverage that would be required? And for example, those PBK models that we use, are they actually sufficiently accurate in terms of their predictions of quantities such as the Cmax? And then when evaluating the toolbox, we don't want to just take into account, for example, animal data that is often used to evaluate different NAMs. We also want to take into consideration, for example, what is the available human safety data, because of course that's more relevant in many cases for what we're trying to be protective of. We also want to explicitly take into consideration both chronic and acute exposure scenarios, because of course that's what we want to be, again, protective of. And also the toolbox is a systemic safety toolbox. So we want to cover off a broad range of systemic toxicities and not necessarily just focusing on one specific endpoint. And then finally, as I mentioned before, we want to understand what's the sort of decision threshold we should be using for saying something is low risk. So that could be a threshold value on this BER, for example. And so we wanted to try and conduct a study that allows us to explore that. So to do that, we took a benchmarking-based approach. So the idea is that you have a series of chemicals and exposure scenarios where you are establishing a ground truth over whether that chemical exposure scenario is low risk or high risk. So that ground truth is coming from existing kind of risk assessment decisions that have been based on traditional methods. So that those would be composed of chemicals, for example, they're used in consumer goods or such as foods or cosmetics that are already considered to be safe. Equally, consumer goods for the high risk, which would be not consumer goods, sorry, but substances that are used at relative exposures that may be causing maybe some form of adversity or side effects that typically are, for example, drugs that where we want to be protective against the effects that they may be causing from a consumer goods perspective. And so the idea is you run through these, generate the data, treating those chemical exposure scenarios as though they were part of an ab initio risk assessment. So pretending you didn't know anything about them, generating the data through the PBK models and the in vitro cell assays, and then calculating the bioactivity exposure ratios, and then seeing essentially what you ended up with. And in particular, if the toolbox was working well, what you would expect to see is that a nice distinction between the low risk and the high risk exposure categories, depending on this bioactivity exposure ratio. So you would hope to see that at some sort of maybe threshold value of the BER, you could start saying, okay, this is where we're seeing the low risk, and we can start saying something is low risk based on that BER threshold. So we divided our sort of approach into two steps. So we had initially a pilot study where essentially we locked down what the workflow and the toolbox actually were, how we would be using them together, and also this process for how we would evaluate it. In particular, we defined what the metrics were for measuring the performance of the toolbox, which I'll get into later. And then we ran through it for a small set of chemicals. I'll describe that in the next few slides. And also we developed a prototype decision model that we would then test further with an extension to that evaluation. So I'll just go through what we got with the pilot study initially. So again, we described, so we took 10 chemicals, as I said already. We assigned different use case scenarios to them and risk classifications for each of those. So many of the chemicals have multiple risk, exposure scenarios associated with them. Caffeine, for example, has 3 of these, one of which is high risk and two of which are low risk. So, for example, with caffeine, you can, of course, have it in coffee quite safely, but then in some instances, people were exposed to very high levels and there were fatalities involved. So the sort of thing that, of course, we want to be protective of. And then we defined, of course, this workflow. So the only thing really to add here beyond what I've already said is that to estimate the bioactivity exposure ratio, we took the plasma Cmax that we get from the PBK models, and then compare that to the minimum point of departure across the different cell assays that we were using. So the reason for doing that would be that it would represent probably the most conservative estimate of bioactivity, and with the hope that it leads to protective safety decisions. So the points of departure estimation, as I say, we had a sort of POD for each technology. So we estimated points of departure for the IP, for the in vitro pharmacological profiling, the cell stress panel. For high throughput transcriptomics, we were testing 3 different cell lines, so MCF7, HepG2, and HepRG. And actually, we then also used 2 different methods to estimate points of departure from that data. So we used one method, which was developed in-house called Bifrost, which essentially pulls out single gene level points of departure. And we also use BMD Express, which is very widely used in the field. And for that, we were generating the pathway level points of departure. And I think Tara will talk a little bit more about these PODs that you get from high throughput transcriptomics in her talk. But generally what we found was the gene level PODs that we got through Bifrost from the high throughput transcriptomics were the lowest, and so the most conservative. If we were using the pathway level PODs, we tended to find that those were much larger than everything else. So if we use just those and the self-stress panel and the in vitro pharmacological profiling, then generally the lowest POD would then instead come from the self-stress panel or the IPP. So in a nutshell, what was the most conservative point of departure to base a BER on really depend a lot on how you were analyzing that high throughput transcriptomics data. So an important factor in terms of how we make decisions based on these sorts of values that we get from the PBK models or the points of departure is really the uncertainty aspect. So here depicted is the point really that often what we get is point estimates from different technologies. So you get maybe a single value representing your Cmax or a single value representing your point of departure. And that would typically be coming from your best estimate based on the available data. And so here, for example, you can see your Cmax is below the POD and therefore your BER is greater than 1. But of course, we know that those will have inherent uncertainties associated with them. So one way to handle this in the field is to think about what is the sort of safety factor you might associate with this. But another way to think about it is in terms of distribution. So you may, if you can quantify the uncertainty that is associated with those values, you can develop distributions which will show the range of plausible values for the Cmax and POD. So here, the height of the distribution represents the level of plausibility for that value. So these are things that you can get from Bayesian statistical models, for example. And so, for example, here what you see is that while for most of those plausible values, the POD is above the Cmax, you can see there is a certain region here where maybe the Cmax is above the POD. And so if you think about things in probabilistic terms, the probability that the true BER based on these distributions is above 1 is not necessarily certain, right? This is just a cartoon, of course, but we wouldn't necessarily put it at 99% or something. So it's just a way of trying to emphasize the point that if you understand these distributions, you can start characterizing that uncertainty and knowing when you need to do more refinement or when you can actually proceed with some degree of certainty about saying an exposure level is low risk. So this is an example of where you've got very low uncertainty for both the Cmax and the POD estimates with respect to each other. You've got a nice wide margin with respect to the bioactivity exposure ratio. In this case, the probability of the true BER being greater than 1 is probably close to 1 in this case. But again, it's just a cartoon. So anyway, so in terms of that initial pilot study, what we did was we at least characterized one of those distributions, so the Cmax uncertainty distribution. So what we did was we took into consideration the fact that the PBK models can be parameterized in a number of different ways. You can parameterize them using in silico only sources. So that's what we call level 1 PBK models. You've got in vitro parameters that you can use to further parameterize those models. So that's what we call level 2. And then in some cases, you might be lucky enough to have some actual clinical data that you can use to further calibrate your models and refine the parameter estimates further. And so here what you see is, as you work through those levels, what we've done is we've collected from the literature measured Cmax values for a range of chemicals and exposure scenarios and compared that to the predicted values and plotted out essentially the error that you get, the relative error, according to the way in which the models were parameterized. And so, of course, as you might expect, as the models are the quality of the parameterization has increased from in silico only parameterization through to using clinical data. The error essentially decreases. But furthermore, what I'd like to emphasize is that we were able to then use these data to develop a Bayesian statistical model of the underlying distribution, so that those distributions then quantifying the uncertainty and the PBK accuracy of the PBK model estimates. And to cut a long story short, using those distributions, we were able to then establish thresholds on the BER values so that we could say, provided the BER was above a certain value, that the point estimate of the BER, that is, was above a certain value, then we could be confident that it was low risk, that exposure was low risk. So, for example, for PBK models that were modeled parameterized using only in silico estimates, that's this level 1, the BER threshold would have to be at least 110. If the PBK level 2 models we use, so you have in vitro data, then it could be 11. And then for level 3, where you have clinical data, then the threshold could be 2.5. And so just to illustrate this a bit further, this is a sort of result that we got from that initial pilot study. So the blue shaded region represents the bioactivity exposure threshold. So essentially, because it's PBK level 2, so that's again the in vitro parameterized models. So anything above approximately 11, a BR of 11 is considered to be low risk in this decision model. And so we overlay there the benchmark, the results from the benchmark chemicals and exposure scenarios. So the blue ones, again, the low risk ones, and then the yellow ones are the high risk ones. And so what we see quite clearly is that all the high risk ones that we identified are correctly identified as not low risk. So none of them are a region falling in this blue area. So we're being 100% protective under that decision model and using the toolbox and associated workflow. Whereas in this case, about a third of the low risk ones are correctly being identified as low risk. And so we could already support those using that approach. If we were to go up to, say, the level 3 PBK estimates, so where you also have clinical data, then the utility would increase actually to almost 70% in that case. So that study essentially locked in those that workflow, toolbox, and decision model. And then we wanted to test it further with an additional set of chemicals. Initially, we went for roughly 40 chemicals and tried to get as many exposure scenarios as we could. A couple of chemicals dropped off the list for various reasons, so that's why it says 38 there now. And one of the things that we really wanted to make sure we did well was the chemical selection with the extended evaluation. So one of the key points around that pilot study, beyond being a small set of chemicals, is also that those chemicals were manually selected. And that manual selection process can sometimes lead to biases in the way in what is actually being selected. So people can tend to navigate towards the extremes that they worry about, or maybe the extremes that they think are extremely low risk. But we want to really cover off the broad range of potential chemical exposure scenarios that we need to be concerned about from a consumer goods perspective. And so to do this chemical selection, we used a randomization-based approach. And this, without going into it in too much detail, this ended up leading us to select a broad range of different chemistries, potencies, and potential toxicity mechanisms from our suite of different chemicals. And then we were able to assign multiple exposure scenarios for each one, in many cases at least. So this is in a nutshell, the result of that extended evaluation. Again, a very similar plot to the one I showed you before. The only thing that has changed between the pilot study and this is essentially just the set of chemicals and exposure scenarios we were considering. So overall, the protectiveness in this case was 93%. So in other words, there's only 3 of the high-risk chemical exposure scenarios we're not able to be protective of in this case. So 2 of those are coming from a warfarin exposure, oral exposure, and one of them from a respiratory sensitization case with triamyletic anhydride. And then we also got a utility value of 24%. So in other words, 5 out of the 21 low-risk scenarios we could support under that decision model because they're landing in this blue region. As we increase across the PBK levels, we found that actually that did lead to a slight increase in protectiveness. So if we try and amalgamate these results together, and just simply say, okay, imagine if we did have clinical data, or we just had in vitro data, and we always take the highest PBK level we actually had available to us, so the most accurate model possible, then we came to a protectiveness value of about 98%. So we're not able to be protective in that case against the trimetallic anhydride still, but nonetheless for everything else we were. And then the utility here in this case is slightly increased to 33% as well. So we're able to support slightly more of these low risk scenarios using that toolbox. But just before I wrap up, I'd just like to emphasize, of course, again, that even if you've got low risk scenarios falling into this white space, we would call those uncertain risk, and therefore in many cases, especially for the ones close to having a BER value of 1, which is this dashed line, you could look to potentially refining those risk assessment decisions further using higher tier tools. So that's almost the end of my talk. Just to wrap up, we've extended the initial evaluation we did to 38 chemicals and approximately 70 exposure scenarios. The protectiveness metric that we came up with varied from 93 to 98%. So overall, a very pleasing result. And we're continuing to explore this further with the US EPA. One of the main activities there is generating data across more cell lines using high throughput transcriptomics and phenotypic profiling, but just more generally trying to increase and get to that point of scientific confidence in other areas, tying into where we can with the work, for example, with Anna van der Zalm, in terms of looking at those validation frameworks that are out there and seeing where the available gaps are in terms of that scientific confidence and what else we need to look at. So some of that is around looking at POD reproducibility, for example, understanding the sensitivity and specificity of different points of departure methods, and looking at the sort of diversity of points of departure we get across different cells, and also how we do the actual cell line selection to get to a good degree of biological diversity in our assays. Just before I finish, of course, I'd like to thank my co-workers who have worked, of course, extremely hard on this project, and also my colleagues at the US EPA who've been instrumental in a lot of this work. And then finally, just thank you for listening. I'll stop there.
Thank you, Alistair. That was a great presentation. And I'm sure it has inspired many questions. But we will ask that you please hold those questions to the end of the presentation. But you can add them to the Q&A module as they come up. And again, just note if they're for a specific speaker. Speaking next will be Dr. Tara Barton-Maclaren. Dr. Barton-Maclaren is the research manager of the Emerging Approaches Unit of Health Canada's Existing Substances Risk Assessment Bureau. She has been contributing to human health risk assessments and methods development under Canada's Chemicals Management Plan since 2007, following the completion of her PhD in reproductive toxicology from McGill University. She's a leader in translational research, bridging innovations in modern toxicology research and human health risk assessment, and serves as the focal point for the development and implementation of new approach methods for chemicals assessment. Dr. Barton-Maclaren is active at the international level as well, leading and contributing to initiatives aiming to replace, reduce, and refine animal toxicity testing, including efforts under the Organization for Economic Cooperation and Development, or OECD, and the Accelerating the Pace of Chemical Risk Assessment, or APCRA. Thank you for being here, Tara, and please go ahead.
Hey, thank you very much for that kind introduction, and to the organizers for inviting me to speak here today. It's really an honor to be part of this epic webinar, and certainly thank you to Alistair, who did a great job in kind of kicking off the session in terms of defining the various acronyms. I know in this area, we get into a bit of an acronym soup, and certainly the concepts. So I'm going to cover, very similar concepts, but coming from a bit of a slightly different lens, if you will. So I work in the existing substances unit at Health Canada. And so we're looking to screen larger inventories of chemicals that are already on the market in Canada, and incorporate bioactivity workflows and other types of new approach methods into our risk assessment activities. The difference is that we're starting, kind of at the top of the funnel compared to what Alistair presented to you, in that we need to use available information and only have maybe the opportunities here and there to generate data to address very specific gaps. So the work that we do in introducing these bioactivity workflow comes from a bit of a different angle, in that we're looking to apply them in a screening context to start out with. So just to kind of give a bit of an intro and comparison between the talks. So I first want to really start out by acknowledging, are the slides moving? There we go. Okay. If they're not moving, please let me know. It was a little slow on my end. So I first want to start by acknowledging all of the fantastic partners and collaborators that we work with and that have significantly contributed to the leadership and success of the various projects that I'm going to highlight today. This includes all the members of my team at Health Canada, as well as our various research and regulatory partners, others in academia, as well as various international organizations. And really acknowledge, as the introduction indicated, that much of this work also leverages work that's ongoing or contributes back to various activities under the OECD and APCRA. So as I mentioned, and as many recognize, the transition to NAM as a complete replacement over animals is likely continue to be a bit of a challenge and a slow process in the near term. And so where we've started under the Chemicals Management Plan in Canada is to ask questions in terms of where can we begin in the near term to find those opportunities to use currently available NAM with respect to our current prioritization and risk assessment needs and activities. And really asking the question and thinking broadly, how can NAM help us to focus the burden of assessing large inventories of chemicals? So as a starting point, we've really focused early developments on application where the program opportunities provide kind of areas or advances where NAM provides information that serves as a step above the current status quo. So we're looking to develop fit for purpose NAM-based approaches to help us make better informed decisions in the absence of other traditional forms of toxicity test data. And this relates to having a number of data poor substances that require screening and possibly future assessment, as well as using this type of information to add mechanistic or mode of action information to inform our assessment activities as well. To support the assessment activities that are ongoing and ensure that we're able to leverage all existing and emerging data and have them continue to be incorporated into the work that we do, we've developed this overarching workflow for data collection, processing, and interpretation. At its foundation, what we're calling the Health Canada Automated Workflow for Prioritization, or HOPPR, really is an integrated computational tool that's built using the NIME analytics workflow. And HOPPR was built to automate 4 particular categories of tasks. First, chemical data collection and storage, data gap filling and predictive modeling. And here you can see it incorporates already a number of new approach methods, including in silico consensus modeling and automated structured-based approaches to support read across. The third task that's automated is evidence evaluation and confidence scoring, which helps to reconcile differences across data types. So for example, there may be higher weighting to a traditional OECD test guideline study versus an in silico prediction. And then finally, it provides hazard and exposure-based prioritization scoring. I'm only going to focus on the hazard indicators in this presentation, but just to highlight that there's also an equivalent exposure arm being developed. And then finally, this provides information reports to support further activities, gives data landscapes, opportunity to identify data gaps, and prioritizing chemicals for further work. The NINE platform is modular by design, allowing flexibility to modify as science and information evolves. And this provides a real opportunity to implement novel approaches, both for the collection, but also for the interpretation of both NAM and traditional data streams. Each of these nodes in the workflow combines various biological levels into a decision framework. And these really are structured in the form of an IATA, which includes everything as available from in silico predictions to guideline type studies. This allows us to really use the best available data and, importantly, to process it efficiently and in a reproducible way. This helps us to deliver transparent and evidence-based decisions through integrated knowledge. And this is particularly important as we start to integrate the various bioactivity and other NAM type data and workflows. Due to the iterative design of this workflow, and obviously the speed at which new and emerging information is becoming available, this really helps us to incorporate the research and new data that's being generated, and this can feed into the process to address various data gaps. It also provides a strong foundation for further collaborative work to continue to refine and improve the tools, as well as the interpretation of the data for use in risk assessment applications as it becomes available. I want to highlight here that we're developing this workflow recognizing that NAM may play a role in both protective or hazard agnostic approaches, as well as working to integrate into more predictive or hazard specific approaches. The start of this journey really did start in terms of the application of bioactivity in a more protective manner, quite similar to what Alistair had presented. And so I'm going to start the journey here. This started with the Health Canada Science Approach document, which built on the APCA retrospective case study that was published and led by Katie Paul Friedman. And that APCA case study really looked at a large group of chemicals to evaluate the use of in vitro bioactivity as a lower bound estimate in a protective way to the in vivo adverse effect levels. We built on this and selected specifically chemicals that had been previously assessed under the chemicals management plan. So we developed this bioactivity approach workflow, which you've already heard quite a bit about. And in this case, we focused it on the ToxCast endpoints using the 5th percentile AC50 values, using in vitro to in vivo extrapolation. And this was the HTTK-R package published by the EPA in order to derive bioactivity points of departure. We looked at this workflow in a parallel way to what the traditional risk assessment inputs require, and to see at the end of the day if comparatively the bioactivity ratio, or the BER, would put us in a consistent place in terms of the level of concern that we observed in the conclusions based on the margin of exposure from our risk assessments. So we compared the bioactivity PODs to the traditional PODs that were extracted from those 42 risk assessments. We then compared them to Canadian exposure values that were extracted from biomonitoring data, environmental media, and consumer products. And then these BERs, as I mentioned, were evaluated to assess the utility of bioactivity data and evaluate the accuracy compared to the risk assessment outcomes in terms of risk assessment. So this is a snapshot, and you'll be used to seeing these plots with various data points on them. So this is published in the bioactivity exposure ratio SIAD, or the science approach document. And what we found was that, in fact, for 38 of the 41 chemicals previously assessed, the NAM-derived AEDs were lower than the traditional points of departure, so indicating that they were protective. So the AEDs in the blue triangles and the traditional points of departure, you can see here by the yellow data points. What was also promising based on this first step was that all the non-genotoxic chemicals assessed as cephatoxic to human health, indicated by the red arrows, fell within kind of this BER range that would indicate potential for concern. And this was similar for the ecotoxic chemicals. What this evaluation did highlight was the potential for outliers. One particular case was indicated here by this purple star, quinoline. It was assessed as potentially genotoxic, but identified using this approach as a low priority. So this, as well as a few other exclusions that we identified in this initial analysis, suggested that, in fact, perhaps a parallel approach focused on screening for genotoxicity was needed that builds on these experiences. This science approach document underwent public comment period, and based on the early feedback that we received, we've really continued to refine various aspects and improve some of the evaluation elements of this workflow. So specific to these ongoing refinements, the beauty of the development and this NIME workflow is, as they're published, we have the opportunity to integrate. And so we've focused on some of these updates being ensuring that we're using the latest versions of ToxCast database. We've updated the assay filtering criteria, more strict criteria, so that we can filter out spurious concentration response curves. We've introduced new functionality to filter out activity that occurs above cytotoxicity thresholds. We've updated again to the latest HTTK-R package. And this circle in the middle is what I'm going to focus on and highlight a little bit more in the case examples that I'll show you. And this really is that we're working to introduce this new ability to move away from the nominal assay concentration in order to be better able to account for the in vitro distribution using the Armitage mass balance model. And we're looking at this to see how much this can improve our AED estimates for these data. When we're doing the HTTK IVIVE, we have typically assumed in the high throughput approach that the in vitro benchmark concentration, or BMC, is in fact equivalent to the freely dissolved concentration and the concentration in the serum blood that exerts a toxic effect. When we're applying the IVMBM, or the in vitro mass balance modeling, this in fact uses assay and chemical specific information to adjust for the in vitro toxicokinetics. And so this calculates the freely dissolved concentration, as well as adjustments for the in vitro to in vivo bioavailability, which corrects differences between the exposure medium and the serum. And so this then adjusts the in vitro point of departure that is used in IVIVE to derive the administer equivalent dose. So this becomes your adjusted point of departure. And I just want to acknowledge that much of this work is being done in collaboration with Arnott Research and Consulting. The case study that I'm going to first highlight really focuses on the two areas that I've just discussed in terms of refinements identified as needed from the SIAD results. So we're looking at the need for an approach to improve the predictive screen and evaluation of genotoxicity, as well as to evaluate the impact that the IVMBM has on refining the IVIVE. This case study initially started under APCRA and very quickly grew into the case study that I'm going to demonstrate under the HESI GTTC committee. So part one of this case study looked specifically at 31 compounds, and these 31 reference compounds were narrowed down from a list of about 300 possible chemicals, and really this was based on them having at least one source of NAM data, so one source of genotoxicity NAM data, and an in vivo point of departure to compare that to. The chemicals that were included had various mechanisms of action for genotoxicity. And the information that was available from the genotoxicity NAMs was evaluated again using benchmark concentration modeling and extracted to human administered equivalent doses using, again, the EPA HTTK R package. The results of these 31 compounds were then compared to the animal-based PODs to see if, in fact, this approach, which was refined and specific to the genotoxicity NAMs, improved the concordance to the animal-based points of departure. Across the 31 chemicals, there were a number of endpoints that could be compared. So in terms of the NAM AEDs, there was 198 that were derived from the in vitro genotoxicity data, and these were compared to 321 PODs from the in vivo data. What we found was that 20 of the 31 chemicals, and this is highlighted in the pink shaded area, had median AEDs that were in fact lower or more protective of the animal-based points of departure. What we did find also in this study, though, is that 11 of these chemicals, the AEDs from the genotoxicity NAM data, were not protective. And so you can see that the in vivo points of departure are in fact lower. In some cases, there's very small differences, but when you look at the bottom of this figure here, the distance between them becomes much larger. And in fact, there's a few of them that had a 1 order of magnitude higher than the median in vivo POD. The same message is really shown here in the histograms, which are displayed as the log 10 differences. And I guess the bottom line here that I wanted to share, that with this improved and genotoxicity specific approach, we're doing a better job and quite a good job at about 60% protective. And deeper into this publication that was published by Dr. Mark Beale at Health Canada, this prediction or this protection can be increased when the analysis was based on the mode of action most of the time. But as a next step, we still wanted to see if there are further refinements that can enhance and further improve the accuracy of the IVIVE and work to minimize the discrepancies between these AEDs and the PODs yet a little bit more. This brings us to part 2 of the case study, where instead of using that nominal in vitro point of departure, which we used in point 1 in the more high throughput way, for the IVIVE, we're then going to examine if these adjusted in vitro PODs using the IV-MBM to account for the in vitro disposition can in fact improve that AED concordance with the in vivo departure. I'm going to take a second on this slide because it's a little bit more complex. So this is the first step, and this figure illustrates the impact of the in vitro mass balance model before undergoing the IVIVE. So we start with the nominal concentration. You then apply the scaling factor to account for the in vitro bioavailability, and finally account for the bioavailability differences between in vitro and in vivo, ending up with this IV POD, or the in vitro POD. Looking over here at the figure, sorry, I'm on the wrong screen. When the IV-MBM scaling factor is applied, the calculated BMCs, which are shown in blue, can be substantially lower than the nominal values illustrated in green. For the bioavailability in plasma, this is shown in the pink. So the bioavailability in the plasma can be greatly reduced in comparison with the serum-free or low serum exposure medium conditions. And what you see here is that for compounds with the higher log KOW, the bioavailability in plasma was lower than the bioavailability in vitro, and this can result in a net increase in the in vitro BMC in comparison to the calculations that do not consider the in vitro disposition. And so you can see in the pink color here that this was a case for a number of the chemicals. And really, again, the take-home message is that this impacts those with a larger KOW more than others. So now the next step is to bring this in vitro POD forward, apply the IVIVE to derive the AEDs, which then can be compared to the nominal AODs from the first part of this case study. What we see here are the results after, as I mentioned, applying the HTTKR package. And here, the point of this step in the study was really just to look for improvements in the AED after applying the adjustments. When we're looking at the AED in the nominal, in this case, the nominal is the pink. You can see that compared to the AED after the adjustments, all of the compounds where the AED originally was lower still is lower than the in vivo points of departure, and the vice versa is true. But what I really want to highlight here, and the key finding, is that we managed to close the gap for 11 of the compounds analyzed. And you can see in this case where the purple diamond becomes closer or better aligned with the in vivo point of departure, which is in the green star. Now, again, at the bottom, we still see these exceptions. And so this highlights that throughout each process and throughout each refinement, there's a need to further establish the domains of applicability, look to see if we can establish exclusion criteria and develop some guidance around these findings so that we can extend these evaluations to a broader chemical space, which is really the goal for the domestic substance list. Without this, it'll be difficult to do that high throughput screening and really identify those chemicals where the further refinements will have the greatest impact. I'm going to shift now to work that's ongoing to advance the use of transcriptomics for chemical assessment. And the main focus here has been on developing approaches or workflows that help us to interpret the large and complex data sets that are being generated using omics technologies. To move to more routine use, there really is a need to establish acceptable practices and work certainly towards best practices on the methodology used to establish the points of departure. And so we've been working a lot to address the question around these acceptable practices and really how do we define what could be considered as an optimal or various different approaches to derive the point of departure. A recent study that we published that strives to get at this answer, if you will, was a meta-analysis of existing data sets where we compiled various published studies, which resulted in data for a total of about 179 concentration response experiments spanning 117 chemicals. As you can see here, these various experiments covered a number of different models as well as exposure conditions, but we did limit to the available information that used the TempoSeq data for kind of one baseline element. This is the workflow on the left-hand side, or the overview of the workflow that was used to derive the in vitro points of departure for comparison with the apical PODs from curated databases listed in step 4. In terms of the processing, back to step 1, we did use the original pre-processed data that were produced from independent bioinformatic pipelines. And then what I'm going to focus on more for this example is really the step 2, or the derivation, where the T-pods were derived again from the in vitro concentration response data using BMC modeling. And this mathematical model helps us to identify the defined response above background. And then these BMCs were used to derive transcriptomic points of departure using various approaches. And in fact, in this meta-analysis, we looked at 7 different approaches to evaluate or to calculate the T-pods falling under two separate tracks. A little bit more detail or more specifically regarding the tracks that we used in this case. So looking at these gene accumulation curves, each chemical is depicted as a distribution of genes with benchmark concentration to derive these T-pods. And we're using the T-pods to define the point of concerted molecular change as indicated here in this left-hand figure. Then taking those various BMCs or T-pods, you can derive the actual effect level or those points of departure based on 2 different approaches. The first, we looked at various distributions to derive the T-pods, and the example on the right shows how the selection can differ between perhaps the 20th lowest BMD value, the 10th percent of the gene BMD values, or it could be the mode of the first peak. We didn't use exactly those, but we did use approaches that are similar in the 5th percentile, first mode, or 25th ranked gene. And then we also use gene sets to derive these T-pods. And this used the median gene BMC value of the lowest or the most sensitive gene set from a selection of various open source curated pathway databases as listed here, KEGG, GO, and Reactome. So taking the 117 chemicals and deriving these T-pods from the various approaches, we were able to do a general comparison across all of these approaches to see if there were any that were emerging as being, more robust than others in a consistent way. Here, this figure shows all of these T-pods compiled into one large figure. I know, again, it's very busy, but really just to hit home the message that really there was overall good agreement between the data points, suggesting that across these various data sets, there was really robust transcriptomics data. These are each subgrouped. Each of these panels are subgrouped by each data set that was evaluated and ranked by their chemical potency based on the T-pods. The blue represents the BMC distribution, and the pink, those T-pods generated using the gene set or the pathway approach. The one thing that did jump out is panel A. So this represents BPA and alternatives. And out of all of the data sets that we evaluated, these came out as being some of the more potent T-pods, but there seemed to be less agreement between the approaches. So some additional evaluation and look into this data set. In terms of the, the other data set, they were all quite well aligned. And one observation that continues to come out in the evaluation is that some of the commonly used approaches may not be the most reliable. And so as I mentioned in the science approach document, we did start with using the 5th percentile, but this seems to be emerging as maybe an overly conservative approach in many cases as we start to expand the evaluation across different types of bioactivity and different types of chemistries and data sets. So the final finding in terms of this general comparison is that we are finding that the 25th ranked gene and the gene set lowest and most sensitive gene set analysis seems to continually emerge as sound alternatives to some of the other commonly used approaches to define that transcriptomics point of departure. We then wanted to extrapolate these PODs again into administered equivalent doses using, again, the HTTK-R package and compare these to the apical PODs. This figure illustrates just that. So we carried forward the lowest and the most conservative estimate from our T-pods and plot them along the apical PODs from the in vivo studies, shown here as a log 10 ratio. What we found is that our NAM-based values, again, are lower or more protective than those estimates from the reproductive, developmental, or chronic dose animal studies that we extracted as comparables. And this was true regardless of whether we were using the BMC distribution approaches as shown in panel A or the gene set approaches as shown in panel B. Again, we're always interested in these learnings. And so we see that these chemicals highlighted in red were exceptions to that rule of protection, where in this case, we actually had traditional points of departure that were lower than the NAM-based PODs. And so looking further into some of these chemicals provides insights on the types of physical properties or the types of chemistries that require further refinements or additional approaches to be incorporated into our workflows. So in these cases, some require metabolic activation, which is true of cyclophosphamide. Some of these chemicals may have multiple routes of exposure and toxicity, or the in vitro cell liner model used just didn't capture the type of toxicity that is elicited by some of these chemicals, such as for the herbicides, for example. So the meta-analysis was really important to suggest that maybe we're not at the point where we can suggest or recommend a single best or a universal T-pod, and that perhaps a range of approaches across the diverse in vitro data sets may need to be considered as we move forward. We wanted to explore how we could integrate this thinking and use all of the bioactivity information that's provided across the various types of cell lines and approaches and integrate these to better support screening and assessment decisions. To do this, we're looking at integrated approaches to testing and assessment, and this represents a flexible framework. It allows us to include a range of different methods and sources of information, as you've seen, are continually being generated and become available. We can assemble these in different ways. And in this way, we can develop these fit for purpose applications so that we can use the various lines of evidence in different regulatory decision-making contexts, really depending on the assessment questions and the protection goals. In this case, the lines of evidence can be integrated for situations such as to support grouping and read across, or they can be all integrated to support overall weight of evidence for assessment. So in the next example, I want to highlight how we're working to integrate different types of approaches and bioactivity points of departure, both from the protective workflows and from the workflows that are being developed in a more predictive way, using endocrine disruption activity as an example. And this example works to incorporate both the bioactivity and biomarker signatures, looking at endocrine mode of action for bisphenols. The development of the IATA approach using a subset of bisphenols really began with the publication of the OECD IATA case study last December. This case study was followed by some further examination of the data set as published in ToxSci, and a study led by Ella Atlas at Health Canada and Carol Yock at Ottawa U. And these two first publications really worked to explore how high throughput transcriptomics could be used to establish mechanistic similarities in data poor BPA alternative chemicals, as well as to compare potencies and continue to increase our understanding of how these lines of evidence can be used to support prioritization and assessment of other data poor chemicals. We've continued to build on these works to pilot a workflow that aims to more generally guide the use of the NAM data in different assessment contexts. And this is the workflow in a paper in preparation that we're working on, which provides the overview of this tier-based assessment workflow that was used to evaluate the different lines of evidence and the different streams of data, and to ultimately determine the estimates that can be used in further risk assessment activities. In the interest of time, I'm not going to walk through each of these steps, but really just highlight that the goal of this tiered and integrated workflow is to illustrate how the various pieces of information from in silico, ToxCast, and the in vitro high throughput transcriptomics can be used in more of a predictive toxicity manner specific to estrogen activity, how they can be used then to estimate a point of departure. And in the cases where you would have data poor chemicals, where through this more predictive workflow mechanism, you weren't able to identify points of departure to derive the applied equivalent doses, introducing the non-specific toxicity stream still has value in that you're able to include potency comparison across all chemicals in a group, even if a specific mode of action is not well understood or identified. So bringing this forward and finally illustrating how really can we put all of these pieces of information or all of these lines of evidence together, we've illustrated at the end in terms of this BER estimation, how non-specific toxicity pathway information, as well as ER-specific information can be integrated and compared in this overall analysis to support the examination and evaluation of a group of both structurally similar and functional alternatives. What we're seeing here again is that the in vitro bioactivity data demonstrated to be quite robust, and we're seeing quite strong agreement across the two different approaches in this case and for this subset of chemicals, where the AEDs for the non-specific toxicity, highlighted in the yellow color, produced in many cases very similar values to the ER-specific estimates when those values were available. And again, kind of that same story resonating across the two presentations is the bioactivity-based AEDs were typically lower than the apical PODs from the animal data. Again, we're highlighting here where some of those typical approaches that you've often seen, such as the lowest pathway, are showing to be overly perhaps conservative compared to these different approaches that we've evaluated, specifically the 25th ranked gene and or the biomarker and ER area under the curve approach from the ToxCast data. So as a final conclusion, in terms of the integrated approach, the use of the various lines of information and the bioactivity estimates can really provide practical information both to inform potency and mode of action for hazard and risk characterization. Learnings and insights, I think it's clear that using these automated workflows allows us to use new information and increasingly complex information to maintain the flexibility to update and ensure that we're using the best available science in an ongoing way, in a transparent, reproducible, and efficient way. There was interesting insights in terms of the application of the IVMBM, where we did see greater impact for the higher log KOW. But on the con side, and this really underscores the need to develop this guidance and maybe further annotation to identify those chemicals for which these refinements are needed, is that it does require specific information that's often difficult to find or not available for the data poor chemicals, and it also reduces throughput if that's a goal of your approach. There are many technical and practical challenges remaining, and just want to highlight, similar to what Alistair had mentioned, this need for characterizing uncertainty. I didn't mention in here, but this also applies to the methods that I've highlighted here. And then the question still stands, if there is a need to have predictive approaches for each of the toxicity endpoints of regulatory interest, I think we're really still touching the tip of the iceberg here. We've demonstrated a lot of valuable and useful approaches to using bioactivity in a protective way, but to get to the predictive approaches using NAM across the various toxicity endpoints, there's much work to be done. Finally, to wrap this up, I want to reiterate the importance of the research and regulatory collaborations. These are truly imperative and really have been the success to where we are at Health Canada to date, and wanted to encourage everybody in this field to continue to build a common vision and maintain that commitment to advance alternative methods to maintain excellence in science-based decision-making. So with that, I thank you very much for attending the session. And yeah, if there's any questions, I guess we're at that point in the session. So thanks, Christy. See, you've popped up. I will try to stop sharing.
No worries. We are indeed at the Q&A portion of our
Okay, I've stopped sharing. Yep. Okay.
Yeah. Yep. Alistair, I don't know if you want to, there you are. So just to orient everyone briefly, we have a Q&A module, which you can reach at the bottom of your screen. Please put your questions in there. We have a few minutes to address them. And also I wanted to tell you that there are a couple of answered questions already by Alistair. So if you check that out, you can see the responses. So the first one is maybe just a really quick one for you, Tara. It's, is the Hopper tool publicly available, or is that an internal Health Canada tool?
So this is currently a tool that is internal. We are drafting this into a science approach document, which we hope to publish, I was going to say kind of early 2024, if that's fair. I think we're aiming for probably end of winter, early spring. And so that will provide the first opportunity for anybody to take a look at the science approach document that really describes Hopper, its contents, some of the decision workflows, etc. So we hope that, you know, eventually it will be something that's more publicly available, and that will be the first opportunity for everybody to take a look at that.
Great. Thank you. So we have a couple of questions about in vitro concentration, so sort of nominal or actual concentrations. And one of them is around, you know, the concern of binding to some of the plastic and some of the materials. And then another one was specific to your presentation, Tara, was asking about the Armitage program, and is that the only one available, or are there other kinds of programs that you can use to help with that understanding? So maybe we'll go to Alistair first, maybe if you want to share a couple thoughts about this topic and within your approach.
I mean, yeah, it's definitely very important. We do initial assessment on whether or not things like plastic binding or volatility would be significant. And so I think for the chemicals that we were looking at, we found that wasn't necessarily the case. In the work I was talking about, we do take nominal concentrations, but what we've also been doing in parallel is generating a large set of data across the same chemicals, comparing the the sort of predictions you get from the Armitage-like models to what you can actually measure in terms of free concentrations. And that's going to be forming the basis of a separate publication where we also look at decision flow, and when you need to maybe conduct further experiments to get a better understanding of what the free concentration is depending on the underlying chemistry. So yeah, definitely very important, and something that we'd probably be looking to integrate into our overall decision framework and toolbox further in the future, I think.
Great. Okay. Tara, what did you want to add?
And then really just to build on that in terms of the question, you know, is the Armitage model the only one available? I think really this is where we have started And as I mentioned, looking to gain experience across broad chemical space and to see if we can develop a framework that allows us to determine the different tiers of refinements that would be needed. And so as part of evaluating this model and then looking to, where it works well or not so well, that will also lead us to explore different opportunities and different methods to incorporate into the workflow. So I think we're kind of at the first steps in exploring these refinements and our ability to incorporate them into this more automated workflow. And in doing that, I think, some of the important lessons or insights is finding out where these models clearly don't work well and why they don't. And so we can better identify those chemicals to do a little bit of a deeper dive, if you will, to see if there are other methods that need further work so that we can, start to incorporate different tools in the toolbox, I guess, going to Alistair's terminology, and when and how do we apply these in the best informed way that we can.
Great. So we have a question about essentially how close are any of the presented methodologies as alternatives to animal testing being adopted by governmental regulatory agencies. Now, Tara, of course, you've talked a little bit about how you're using these approaches. I don't know if you want to speak to whether you consider them alternatives to animals, or if you're thinking about integrating them, more broadly into decision making, and then we can go to Alistair.
Yeah, I think in terms of, exact timing, it's very difficult to say because there is a certain level of readiness that corresponds with the context of use. And so in terms of readiness and some of the prioritization workflows that I've illustrated to you, we are using them. We are using them in the context of prioritization. Certainly, we're looking at the bioactivity and the BERs to help us prioritize chemicals that would have otherwise not been prioritized, perhaps because of a lack of traditional data. So in these contexts, we're currently using them. In terms of using them in formal risk assessment and decision making, we have used NAMs for quite some time in this aspect, read across, for example, and we continue to integrate this new information with traditional sources to support the risk assessments. We're not using them as standalone or replacements at this time in many cases. Skin sensitization, the defined approach may be an exception to that in our new substances program where we are seeing submissions. But it's going to be an iterative and progressive approach, I guess, to integrating as the approach becomes available within the risk assessments. And again, kind of hitting on the data availability in our program, it's in existing substances. It's often difficult to integrate if the data is not available for the chemicals of interest. So that's a lens. There's kind of two prongs. Is the method ready? And is the data available?
Yeah. And Alistair, you want to?
Yeah, no, I mean, the only thing I would say is I guess there are a number of examples in this field where, NAMs have been shown to be sufficiently predictive of specific endpoints and therefore have been accepted from a regulatory point of view for use in sort of toxicity assessments. I guess the challenge that we have in this space is that we're moving more to a protective viewpoint and how we use them. Without necessarily being predictive, and the key point is really integrating the exposure and the hazard information to make the safety assessment. And so from a regulatory point of view, moving to a space where the tools can be evaluated and validated within the context of exposure, I think from what we're trying to do is a key challenge. So that's all I have to say on that.
We have a question for both of you, but I want to put it to Alistair first. It's about cell line selection, and you presented a lot of information. So maybe the cases, it's kind of a case by case basis, but they're wondering if the cell lines are selected specific for chemical modes of action, or are there some other process?
Yeah, no, so for the, well, particularly, I guess this is relevant for the transcriptomics analysis that we do. Yeah, so for those, we did select what we thought were 3 reasonable cell lines initially, to just try and cover off a broad range of potential bioactivity. But what we've been doing more in a sort of more systematic way with Josh Harrell at the US EPA is coming up with, well, it's really Josh's work. It's looking at the diversity of baseline expression across a broad range of different cell models, and then basically having a process for selecting those cell models based on biological diversity of the baseline expression. So that allowing you to then try and cover off a broad degree of biological space with really as few cells as possible. And so that's just at the end, my final slide, I just show a brief snapshot of some of that data that we're generating, where we then selected those cells and then treated them with the different chemicals to see what we get out. I mean, it does turn out that actually the 3 cells that we'd selected were among actually some of the most diverse that he had actually pulled out in his analysis, but that was after the fact, actually. But yeah, I think generally speaking, I mean, again, going forward, we want to be able to, of course, select a good range of biological diversity. And I think, for example, Josh's work in terms of having a process for doing that would be really important in that space.
Yeah. Tara, did you want to add anything?
No, not a lot to add. Really, we're working with the EPA and the folks that Alistair mentioned as well. And there's a case study under APCRA looking prospectively, and again, looking at some of the questions around the selection of cell lines and how they can be improved to be more predictive of certain endpoints of toxicity. So just to say, we're kind of working with the same folks on addressing that question. I just see a question here that I did want to answer quickly in terms of, endpoints that are not included in the predictive section. This is kind of Hopper 1.0 as we have it. And so that's the beauty of this workflow, is that we will continue to expand and incorporate other types of toxicity. Just in this version, we limited to those that are most commonly used and where the data most frequently exists in the context of the chemicals that we evaluate. But certainly we're looking to expand and continue to incorporate all types of information in more nodes in iterative versions of the tool.
Great, thank you. So I'm so sorry we are out of time for questions. Thank you to everyone who added your questions, to all of you who listened today, and in particular, thank you to Tara and Alistair for your presentations. Thank you also to our co-hosts, EPA and California EPA, and the PETA Science Consortium. We will be posting the recording and the slides for the webinar on the PETA Science Consortium International website very shortly, in the next couple of days. And please look forward to our next EPIC webinar, which is going to be in February. Thanks again to both of you, and thanks for joining us, and have a great rest of your day.
Thank you very much.
Yes,
Thank you everyone
thanks
for listening.
everyone.
Yeah.
Nice to see you.
Nice to see you all. Bye-bye.
Bye.